Hi, this is Dr. Lonneville, and we're going to talk about Western blots. Western blots are concerned with separating protein and looking for a particular protein among a mix of proteins. So you have to extract protein from your tissue, whatever your sample is going to be. In our case, we're going to extract from worms, and I'm going to show you how to do that. It sounds fancy, but it's not that fancy. Basically, we're just going to wash some worms off a plate and grind them up. For other situations where you're doing a Western blot, you may have already a preparation of protein. Let's say you're separating cells into different organelles. For example, you're doing a ribosome prep, or you're doing a mitochondrial prep, or something like that. You need to know what the protein concentration is. Or you're doing some kind of protein purification and you want to know at different fractions of the purification procedure, how much protein have you got. You need a sample of protein, and that can be very crude, where you're just taking an organism and grinding it up and looking at the total protein in that ground up organism. It has other stuff in it too, like fat, nucleic acid, carbohydrate, but you're ignoring that and just looking at the protein. So what we're going to do is just wash off some worms and grind them up, as you would most likely do for a Western blot that you're going to do for your independent project. Okay, I have a plate of worms that I've been growing, and I checked under the scope that there are live worms on there, and I'm just going to pour a few milliliters of S media. So this is liquid media that you could grow worms in if you supplanted it with bacteria. I'm just going to wash that around a little bit. Okay, so I have let that incubate for a little bit on that tube, just a little bit, like a minute. I'm just going to now swirl this fluid around, use a plastic pipetter and remove that fluid and put it into a Falcon tube. I'll show you that in a second. It's off camera. So you can wash plates like this and it will remove quite a few worms from the plate, but not everything. You can wash these plates and then still keep the plates in the tube, in the incubator. Off the plate now, and it's in a Falcon tube, and you can see that it's pretty cloudy, and those are worms that are going to settle. So I'm just going to let it sit upright now for about 15 minutes and just let the worms settle. You can centrifuge them, and some protocols say that you centrifuge them and then you don't have to wait the 15 minutes. But even at gentle speeds, our experience is that ends up killing the worms, and we would like the worms to be alive for this particular application, and then we're going to grind them up to release the proteins. The problem is, as soon as your worms die, the proteins can start to denature, and we want them to denature in the presence of SDS. Okay, it's been a bit. I just want to show you what a pellet looks like of worms after you've let them settle. Hopefully you can see that. It's a bit of a glare, but hopefully you can see that there's a brownish, reddish pellet in the bottom there. I'm going to remove that using just a pipette. Okay, I am squeezing the pipette before I go into the tube. So I squeeze it, then I go into the tube, and then I release it to suck it up. If I go into the tube and then squeeze it, I blow bubbles like this, and that's going to disturb the pellet. Okay? So now I've got a small volume, hopefully highly concentrated with worms. I can always put a drop on a slide on the microscope to check for that. And then I'm going to grind them up with a sonicator, which I'll show you next. What I have right here is called a sonicator, or a sonic dismembranator. What it does is it generates sound at a given frequency at this thing right here. That's called the horn. and you can use that to blow apart cells, disrupt cells. This is often used in other applications to sterilize things because it will kill bacteria, and it's a common homogenization technique. The reason we didn't do this when we isolated RNA was this is an expensive thing. It's like $5,000, and there's no way we could be sure that we could clean this well enough that there was no leftover RNA from one sample to the next. And so that's why we use the beads, because they're disposable. In this case, since we're dealing with protein, it's much less worrisome about contamination if we just wipe off the tip between runs, then we're okay. So what you're going to hear is a high-pitched sound. All I'm doing is flicking a switch. I have them in a little container which will keep them cold, because this process, the sound waves generate heat. Heat is the enemy of proteins. It'll denature the proteins. We don't want them to denature until we say so, in the presence of SDS. So for this point, we're just trying to open up the worms, get the protein out, and you'll hear this high-pitched whine, and then I'll go on to the next sample. The horn in the bottom of the tube, I'll count to 3. 1, 2, 3. Okay, now I'll wipe off the tip. For the next sample, 1, 2, 3. Wipe off the tip, and then that's it. Okay, so I have done a protein assay where I put no protein in there, 2 micrograms of BSA, 4 micrograms of BSA, 8 micrograms of BSA, 10 micrograms of BSA, and then I measured the absorbance at 595 for all of those values, put those in here. And then, as you remember from Lab 1, you say tools, data analysis, regression, okay. My Y values are the absorbance values, and my X values are the protein values. And then I'm going to check all these boxes down here. I say okay. And I get a regression. I want to take a look at that regression. The orange boxes are a perfect straight line, and the blue boxes are my data. If I zoom in here, values. So the R-squared value is 0.967, and that's pretty good. A perfect straight line would be 1, and anything above 0.9 is acceptable. That means I can explain 96.7% of the variation that I see in my absorbance with a straight line. So that's pretty good. And then down here, my intercept is 0.169. That's where it crosses the y-axis, and my slope is 0.096. So then if I go back to my data sheet here, I can just plug that into a formula. Up here, I'm saying my absorbance value here minus 1.169 divided by 0.097. So I'm just solving for X to find out how much protein is in my unknown. So my first sample of unknown, it says there's 4.237 micrograms of protein, and then 3.92 in my second replicate, and then the second sample 4.95 and 5.16. But that came from 2 microliters of the sample. So if I want to know how many micrograms in 1 microliter, I just divide this by 2, and I get these values. Bottom line is sample 1 looks like it's about 2 micrograms per microliter. Sample 2 looks like it's about 2.5 micrograms per microliter, and I'm going to use these data to make my samples, which I will do next. Okay, so now let's make up some samples for the gels. The sample buffer contains 4x sample buffer plus your protein sample plus dithiothiotol plus water for that's the total sample. So in the 4X buffer you have, and you have recipes for all of this on your protocol, you have dye, so you can track your samples and track the electrophoresis. You have the SDS, which will unfold your proteins and coat it with a negative charge. You have some glycerol so that the samples become viscous and they fall to the bottom of your well. And then some tris buffer. And your proteins came from the worms. That's we just figured out what the protein concentration is. DTT, dithiothiotol, is a reducer of disulfide bonds. So it breaks covalent bonds between cysteine residues, and that's important for completely unfolding the protein. We'll talk a bit more about that later in the video. And then finally water, so that all of your samples have the same volume and you're loading the same volume into all your wells. Okay, so there are some things in each sample that aren't going to be the same no matter what. So first you got to figure out how much are you going to load per lane. Okay, and we're saying that we're going to load 20 microliters per lane. You can get more than that in there, but for people who haven't run a lot of Western blots, 20 microliters, you should be able to get that in the lane without spilling over into the next lane. Once you get more practiced at it, you can go up to more like 30 microliters per lane to get more in there. And then how much protein per lane. So you can load on this format of gel, if you have a complex sample of a protein, anywhere up to about 50 micrograms of protein. So a complex mix of proteins. What does that mean? That means that you're grinding up a sample that has no one protein that is super overrepresented. So for example, if I took blood out of some kind of mammal, that most of the protein in that sample is going to be hemoglobin. Over 90% will be hemoglobin. And you can't load 25 micrograms of a single protein on the gel. That protein will just show up as a huge blob. But if you have a complicated mix of proteins, like in a worm, has maybe 100,000, 200,000 different proteins in there, no one protein is overrepresented compared to all the other ones. And so each protein in that total 25 micrograms or 50 micrograms is going to be relatively a small amount compared to all the others. And so you can separate them out on a gel. So if these are things you have to consider depending on the tissue. So like if you're working on something that has a lot of muscle and you're working on muscle, well, muscle is dominated by actin and myosin, and so you can't load as much total protein on there because there's two proteins that will dominate your lane. So for the purposes of this lab, worms are complex. You're grinding up the whole worm. You're not dissecting out those worms. You just do the protein assay on those ground up whole worms like we just did, and you figure out how much can I load per lane. And you want to load as much as you can, because the more total protein you add, the more chance that you will see your protein. So let's say you have a really sensitive antibody, and your antibody can detect 1/10 of a nanogram of your protein. Then if you load small amount of total protein on there, the chance that your protein as an individual band on that lane is at least 1/10 of a nanogram. That's pretty high. But if your antibody is not so sensitive, the more protein you load, the better chance your antibody gets to detect that protein. So bottom line is load as much as you can. Today, for this example, we're going to load 25 micrograms of total protein per lane, okay? Not per gel, but per lane. And we're going to make up today 3 lanes worth of sample, okay? I'm actually going to load just 2 lanes of each sample, 2 lanes of sample 1, 2 lanes of sample 2. So why am I making up 3? That's because when you're pipetting and when you're boiling these things, it's easy to lose just a little bit of your volume when you do that. And the most important thing in a Western is that everybody is even Steven. So if I had 3 lanes worth of sample and I loaded all 3 lanes, I would load 20 microliters in the first lane, 20 microliters in the second lane, and in the third lane, I might only have like 17 or 18 microliters. And that means that third lane is going to get short shifted, and so I'm not going to even load that third lane. So by making 3 lanes and planning on only loading 2 lanes, I'll have some slop left over, and I'm sure that those 2 lanes will be equally loaded because I'm sure I can get at least 2 samples of 20 microliters out of there. Okay, so 3 lanes worth of sample, and 3 samples, that's 60 microliters total. 20 plus 20 plus 20, 20 per lane, 3 lanes, 60 microliters. Okay. I want 25 micrograms of total protein per lane, 3 lanes, 25 times 3 is 75. Okay, now I have the information I need in order to make up a sample. All right, over here, here's the stuff that's going to go into my sample. 4X, your protein, DTT, and water. 4X is called 4X because it is 1/4 of your total. So as soon as you figured out what your total volume is, 60 microliters down here, then you know how much 4X it is, 1/4 of 60, or 15 microliters, okay? The next thing you know for sure is DTT. So DTT is a disulfide bond breaker. It should be in all your samples. It's a little stinky. We have a 1 molar stock that we use. We dilute that to 50 millimolar final concentration. So it's 1/20th of the total. So if the total is 60 divided by 20 is 3. So I've got 15 in there and 15 of 4X and 3 of DTT, regardless of what my protein concentration is. Okay? I have to have 4X in there. I have to have DTT in there. I don't have to have water in there. It could be all protein. The water is only added there so all the samples have the same volume when I'm all done. Okay, my sample 1, if you remember, was 2 micrograms per microliter. And I want a total of 75 micrograms. If I divide 75 by 2 micrograms per microliter, I need 37.5 microliters of that protein sample. I'm going to round it up to 38 in order so that every lane will have 25 micrograms. Okay? So this is 3 lanes worth. I need a total of 75 micrograms for 3 lanes worth of stuff. 75 micrograms, how am I going to get that? Well, the sample of worms is 2 micrograms per microliter. I figured that with the protein assay. 75 divided by 2 is 37.5. I'm rounding it up to 38. So then 15 plus 38 plus 3, 4 microliters are left in order to come up with a total of 60. So I'm going to take a tube with those caps that don't pop off. I'm going to put all these 4 things together, and I'm going to boil it for 10 minutes to let the SDS and the heat and the DTT do their magic and unfold those proteins. So I've put together the sample components, the DTT, the water, protein, in these lock and cap tubes, because what we're going to do is we're going to put them in this heat block in the hood, and they're going to boil. They're going to generate pressure, and if you don't use the locking cap tube, they'll pop and you'll lose your sample. One thing I do want to mention is when you're pipetting the 4X has glycerol in it, and so it's very viscous, and you're going to pipette in this case 15 microliters. Okay, so you just want to pipette slowly. It also has lots of bromophenol blue in it, which is caustic, and you don't want to get it all over the barrel of the pipetter. Okay, and you want to wear gloves because that can happen. You get blue stain on yourself, and the bromophenol blue is kind of caustic. So I'm going to add 4X. I'm going to make sure that that lid is on there really well. Okay, and I'm going to stick it in the heat block and let it boil for 10 minutes. Okay, let's talk about gel boxes for a bit. This is an Agarose gel box. You've already used this. A tray that acts like a mold for the gel. You put the mold in the gel like so. Pour the gel, and pull the gel out, and then run to the red in a horizontal fashion. Western blots, they're run in a vertical manner, and this would be your most typical Western blot box. So this is considered a mini gel, and you put a gel up against this green gasket, put another one on this side. See that little notch there? There's a short plate and a tall plate. The gel is in between the short and the tall plate. You smash those two together like this with a clamp. So you put the two gels in there with a clamp, and they're held together on that gasket. Then there's buffer on the inside and buffer on the outside. You can see an electrode right there, that wire right here, and then another one on the outside. Same thing, cathode, anode, run to the red, all that stuff. See, there's a red pole and a black pole. Those are a little easier to understand in a simpler setup. Here's one I made out of Legos. There's nothing really fancy about these sort of things. The gel goes up against this gasket. It sits in a tray of running buffer down here. There's an electrode down there that I added. The electrode's wire is expensive because it's platinum wire, but it's really thin. It's not that expensive. And then another electrode up here. And so this would be the positive electrode here, the negative electrode here. You load your samples up here in the gel. They run down the gel toward the positive electrode, the one that attracts anions. So the anode is the one that attracts anions, and all the proteins are anions because they're coated with SDS, and that's how the proteins separate. And I don't really know why there are no horizontal protein gel electrophoresis rigs. I can't think of a reason theoretically why there couldn't be, but there just isn't. So vertical is almost always for acrylamide gels. It probably has to do with how the gels are prepared and how they are poured versus the Agarose gels. Agarose gels horizontal. Here's another one that I made out of plastic. Again, you can do this much cheaper. So this sort of thing is the cheapest one. You can make one of these for about $10. The Legos are the most expensive part of this one, but it does work. Whereas this from Biorad, which is like the workhorse that everybody uses, is more like $600. But there's all kinds of designs out there, and really how they differ is their propensity to leak. So the problem is that you're creating an upper buffer chamber, like here, and a lower buffer chamber, like down here. And if the upper leaks into the lower through this gasket, then you'll have problems with your electrophoresis. The Biorad gasket, that green gasket there, is one of the most reliable, but there's a bunch of other companies out there. We're going to use an even more fail-safe sort of setup. This is called a Criterion system, also from Biorad, and this one uses what are called precast gels, and all you do is you pop the gel in there. and you add buffer to the bottom and then buffer to the top in a chamber that's already welded to the tray. So this is the tray that you're going to use at least this year, and it's a pre-cast gel. Here's the gel that's in there. We'll get that out and when we load our gels in just a second. These are excellent in that they're very reliable. You can stick them in the fridge and have a bunch of them in the fridge, like a bunch of frozen burritos, and you just take them out when you need them. The problem is they're really expensive. So this thing with shipping is between $15 and $20. You can make your own, that you pour your own in a lab with a mold and liquid acrylamide. You can do that for about, I'd say, between 25 and 50 cents per gel. The difference is that there's some proprietary chemistry in these precast gels that allows them to have a long shelf life, up to a year, and the ones that you make yourself are good for, at the most, a couple weeks. So you have more labor involved if you're doing them yourself. But if you're doing a lot of Westerns, pouring your own is definitely the way to go. If you get good at it, pouring your own is actually superior in terms of the resolution you get versus one that you would purchase. But for students learning how to run Westerns, really can't beat a precast gel. Okay, now I'm going to get ready to set up my gel and run it. So this is how the gel comes. These are precast gels, and this tab on the bottom, super important that you take that off, otherwise you'll get no electrophoresis. So the current is going to run from the samples up here down through that little notch that I just created by taking that tape off. They put the tape on there to allow them to pour the gel, but then you take it off as a customer. And then here's the comb, okay? Each one of those teeth there is where you're going to put your sample. You're going to take that comb out, okay? Just gently remove the comb. Okay, so this is super easy. It just fits right in there like that. Okay, see the numbers there, and those are your individual wells. That's another reason why I like these precast gels for classes and teaching people how to do Westerns, is that it makes it much easier to see the wells. Otherwise, all that is clear and there's no labeling when you pour your own gels, and that's just easier to do after you've done a few. So if we look at this gel here, it's the gel part is between those two pieces of plastic. So the longer piece of plastic closer to the camera, and then the shorter piece of plastic, and then hopefully you can see those individual wells. That's where the gel actually is in between those two pieces of plastic. And then over here, there is a spacer in between the two pieces of plastic that governs how thick the gel is. These are 1 millimeter gels. You can adjust the thickness of your gels to improve resolution. The thinner they are, the better resolution they are, but the thicker they are, the more protein they will hold. So I'm going to add 1X SDS running buffer into the top chamber here, and then the bottom chamber, and you will see on the side here it says fill, and then there's a line there. Okay, so to get electrophoresis, you just need it to be filled to the point where I pulled off that tape near the bottom of the gel. Okay, there's the fill line. And again, in order for electrophoresis to happen, I only need to fill to about there. But the reason that you fill up to here is to act as a heat sink. So electrophoresis generates resistance in the gel that generates heat, and heat affects how electrophoresis happens. And if the heat isn't completely even across the gel won't run evenly, won't run flat. Some samples will run faster than other samples will. These are called smile effects or frown effects. You get a smile if you have increased resistance on the edge of your gel. And if you have decreased resistance on the edge of your gel, you get a frown sort of pattern. Those are no good because you're just assuming that every sample is migrating at the same rate. And so by filling up your buffer, you can provide a heat sink to get rid of any heat that is produced by the electrophoresis. Some people go so far as to stick a block of ice in their buffer. It's a big, long tip, and the end is very thin. It fits between the two spaces. So the first thing I'm going to do is I'm going to load the pre-stain standards. We'll talk more about what that means in the next part of the video. Whenever you are loading up a sample that has got glycerol in it, and that includes all electrophoresis samples, you want to pipette very slowly, especially in these tips that are very, very thin, because it's going to take a while for that stuff to make it up into the pipette tip. If you pipette too slow or too fast and you pull that out, you're going to get a bubble at the end of your tip, which is going to mean you're not pipetting the right amount. So for the pre-stain standards, I'm pipetting 10 microliters, okay? And I'm going to go in front of my sample. I'm aiming for number 1. And then I'm going to come up against the bottom here. I'm going to drag the tip up until I feel a click, like there. That was just it. So I'm going to drag the tip up until I hear a click. See that click? Okay? And then I will put the tip down just about a third of the way in and very slowly pipette that out. And you will see the sample falls to the bottom of the well. Okay, I'm going to very slowly let that out. Sample falls to the bottom of the well. Okay, now instead of going to the second stop, I'm going to pull my tip out like that, and then I will let it go. If you go to the second stop, you can just blow your sample out into the next well and contaminate the next well. Now I'm going to load my first sample, sample 1. Okay, so I'm not just letting go on that plunger. I am very slowly releasing that plunger, giving a chance for that sample to make it into the tip. And this is 20 microliters. Remember, we talked about that. The pre-stain standards was 10 microliters. I'm going to load this next door here in 2. Hear the little click. I'm going to let it go nice and slow. Slowly fill up that well. If I just totally let it go out of that well very fast, it'll overflow the well and contaminate sample 1, lane 1. Okay, you see a little bend in the well there. That's from when I pulled out the comb. I pulled out a little too fast and it left a little bend there. It should run fine. And I'd like to say that I did that on purpose just so that I could show you that that might happen, but that would be a lie. If you notice that you have, here I'm loading this last sample 3. If you notice you have a bend in your well before you start loading samples, then you can try to straighten it out with an empty pipette tip before you load the samples, but I did not notice that. So here I go for lane 3. Click over the edge there, very slowly letting it out. It's going to fill up lane 3. Lane 3 looks like it's not bent, like it's straight. And then I'm going to load the same thing in lane. I'm going to skip a couple lanes. I'm going to load 6, 7, 8, and then I'm going to start the electrophoresis. Okay, I've got my samples loaded. Now I'm ready to start electrophoresis. I've got a red pole over here, a black pole over here. I'm going to put my lid on red to red, black to black, and they are set up so it's really hard to do it backwards. Back here on my power supply, red to red, black to black. 200 volts is what you want for protein gels. 150 for the Agros gels that you did before. 200 for the protein gels. The protein gels are acrylamide. They're tougher. They're not going to melt. Okay, you can see bubbles coming off that anode. So if we look at these samples, you can see that they are flattening out and they are moving into the gel. The gel starts at the bottom of where, like, if you see that lane 4, the bottom of that rectangle, that line there is where the gel starts. So lanes 1, 2, and 3, 6, 7, 8 are starting. If you had hooked up your electrodes backwards, they would move backwards out of the gel. I have done maybe 1,500 of these, and I've made that mistake more than once, so you don't need to make it. So we're going to revisit this gel as it's running, so you can see. Okay, let's talk a little bit about what is happening to your samples when you prepare them. And I apologize for the glare here. So your proteins have a shape, have a 3-dimensional shape, as I have expertly drawn here. When you add soap, that's the SDS, sodium dodecyl sulfate, ammonium lauryl sulfate, ammonium dodecyl sulfate. Look at your shampoo and you will see these ingredients, the same ingredients that are in your SDS page. So SDS is the molecule sodium dodecyl sulfate, which is essentially soap. If you add that molecule plus heat plus the molecule dithiothiotol to a folded up protein, what happens is the protein unfolds into a more or less linear shape, and it gets coated with these negatively charged SDS molecules. Okay, SDS is an amphipathic molecule. It has both hydrophobic properties and hydrophilic properties, and it will interact with both hydrophilic and hydrophobic amino acids to unfold that protein. Okay, what SDS and heat won't do is they won't break a covalent bond, but they'll break all the other kinds of bonds, ionic bonds, van der Waals interactions, hydrophobic interactions, that sort of thing. So if there is a covalent bond within a protein, that's not going to break it. The type of bonds that would be within a protein that affect its shape are cysteine-cysteine bonds, disulfide bonds. And DTT is in your sample buffer in order to break those bonds. It's stinky. If you've ever had parents have ever gotten one, you know that the chemicals they use are really stinky. It's the same chemicals. And what they're doing is they're breaking disulfide bonds in the keratin in your hair to stretch it out into more linear shape, and then you can make it take a new shape with heat, curlers, that sort of. What happens is in your sample, you've got all kinds of different proteins in there. Myoglobin, tubulin, actin, parvalbumin, GAP, DH, etc. All the proteins you guys have read about for your projects. Those proteins are all becoming linear in shape and coated with SDS. Any protein that has a disulfide bond, that disulfide bond will get broken. But what is not happening is there is no dye that is attaching to your sample in a covalent manner. So your samples don't have a covalent dye attached to them. The dye that's in the sample that looks blue is just a way to track the electrophoresis happening. So it's a dye that is responding to the difference in voltage, and it's moving according to that difference in voltage, but it's not attached to any protein. In fact, it runs ahead of the protein. So it allows you to watch how the electrophoresis is proceeding, but it is not the protein. So each year people freak out and they say, oh, my dye is near the bottom, my protein's going to run off. That is not your protein, it's running in front of your protein. In the pre-stain standards, it's a different situation. What chemists have done is they've taken a known protein like tubulin, or like myoglobin, or like parvalbumin, proteins of known molecular weight. And what they did is attach a dye to them with a covalent bond. So green is attached to parvalbumin with a covalent bond, blue is attached to myoglobin with a covalent bond, orange is attached to tubulin with a covalent bond. Therefore, when you separate those and treat them with SDS and separate them by electrophoresis, the dye stays with the protein, and you can actually see that dye separate in the gel. Okay, during electrophoresis, what is happening is you will see in your pre-stain standards, you'll see those proteins separating, because you can see the protein label, which is a dye, and you can see that with your eye, and you can see them separate because the proteins they're attached to are separating. And the disulfide, the DTT only breaks covalent bonds that are disulfide bonds. These covalent bonds aren't disulfide bonds. So those dyes stay on the proteins. But your proteins from the worms that you ground up today, they are not labeled. And you will see nothing while it's running, okay? You will see a dye front that is blue, that is in all the samples. That's just bromophenol blue that's in the 4X dye. That's not attached to your protein, is running ahead of your protein. It's just a way for you to see your samples when you load them up here, and then to watch the electrophoresis progress. That's all it's there for. It is not the protein, okay? This is not labeling your protein. These are actually labeled protein in your pre-stain standards. Pre-stain standards, when you transfer it onto the PVDF, onto the blot, the pre-stain standards come with it because they're covalently attached to the protein. And the protein's going to move, the dye is going to move with the protein. The other proteins are also moving, but you can't see them because they aren't labeled, okay? So the pre-stain standards is just a way to prove to yourself, yes, transfer happened over onto this blot. It is possible to transfer your proteins all the way through the blot, and so you get usually a little bit of pre-stain standards on both sides, but it's brighter on one side than another side, and that gives you an idea. This side is the surface where the protein is. We are checking the progress of the electrophoresis. You can see 2 lanes of pre-stain standards, one right here, one right here. You can see a red marker, a couple blue, and then a green, and then so on. This blue line here and here, that's the dye front. Again, that's not protein, that's running ahead. Okay, we can see that the electrophoresis has progressed well. Those pre-stain standards have separated well, and you can see the dye front there. Pretty soon that dye front is going to start migrating out of the gel. One thing I haven't mentioned before is about the percentage of acrylamide gels. You would gauge your percentage of gel according to what your target protein is. And you want that target protein to be someplace around the middle of the gel, maybe as low as you can change the percentage of an acrylamide gel just like you can change the percentage of an agarose gel, and higher percentage means smaller pores and separates smaller proteins. For protein gels, acrylamide gels, on the low end of percentage is about 4%, and that's for very large proteins, say above 200,000 Daltons, 200 kilodaltons. And the highest percentage gel, which is 20% gel, is for the smallest proteins, say 10 or 10 to 12 kilodaltons. If you have smaller proteins than that, you would switch to a different SDS page chemistry. For really small proteins and peptides, you use a chemistry called trypsin gels, and so on. But the bottom line is that you have to know where your protein is in terms of molecular weight, and you pick a gel that is going to resolve that molecular weight really well. These gels that we're using right here are a gradient of 4 to 20%, and so they separate a complex mix of proteins well. I bought these ahead of time because no matter what protein you pick for your project, these should work.